Thank you very much. Good afternoon. As we're a public company listed on the NASDAQ, so the forward-looking statement. BrainStorm at a glance. We're a clinical stage biotechnology company. Potential to be the first-in-class autologous stem cell therapy for ALS and other neurodegenerative diseases. Experienced U.S. management team in the U.S. and the R&D in Israel. And we have a robust IP portfolio. An advanced stage clinical programs are the repeat dose phase 3 ALS trial, fully enrolled. And repeat dose MS trial in phase 2. And I will give some more detail about this in the coming slides. Of course, a large addressable markets, ALS, 30,000 patients in the US, 450,000 patients worldwide, and unmet need. Progressive MS, even though the MS space is quite crowded with many products, but for progressive MS is an unmet need. And there's 500,000 patients in the US only of progressive MS. It's about 50% of the MS population end up being progressive MS patients. And of course, we know the neurodegenerative disease prevalence expected to increase as population ages. So this is just a glance at our pipeline. As we just said, the 2 clinical trials on ALS and progressive MS, and preclinical published papers on Parkinson's, autism, Huntington's, peripheral nerve injury. NurOwn, we believe, is a platform that has the potential to treat unmet need in the CNS space. So as you can see, Parkinson's, progressive MS, autism, Huntington's. Our lead is, of course, ALS. What's the product? So it's an autologous stem cell product. Until a few years ago, the industry thought that it's not viable, it's not economical. And we're very fortunate that in a different industry within the healthcare industry, in the CAR-T industry, for oncology, all CAR-T cell treatments today are autologous treatments. And they're approved products, 4 to 5 approved products. Novartis has an approved product. Kite Pharma, Juno, and Bluebird. And all of them are autologous cells that are gene manipulated. And even after approval, only 50% of the patients respond, 40 to 50%. And it's viable, it's economical, it's working already in many countries. They're having their issues with the gene modification or the vector issues. Our product is far simpler, it's CMC, no gene modification. It's just bone marrow-derived cells. I'm sure there's a pointer here. No. So we can look at one. The patient comes in only once, where we do the bone marrow aspiration. And the bone marrow aspiration once is good enough for treatment for years to come. So the bone marrow aspiration is a short procedure. Every good medical center knows how to do that. Then we have an intermediate product that takes us 12 days, where we differentiate and expand the mesenchymal cells. We purify the mesenchymal cells, over 90% purification. And we're able to have a cryopreserved, validated intermediate product. And that cryopreserved product, from defreezing and differentiating and expanding, it's an only 7 days process. So it's the closest you will get an autologous treatment to off-the-shelf, to allogeneic. And the truth is the patient doesn't know. He feels only the part of once bone marrow aspiration. Then, whenever the doctor will have a Gantt of the months when he wants to inject the patient, we will be able to provide the same day or a day before fresh cells in a syringe, 125 million cells that are injected intrathecally, again, a very simple procedure. So from 2007 to 2010, we were very busy with the preclinical mainly. In 2010, we started our first clinical trials, the open label trials in phase 1-2, which was published in JAMA Neurology. And in 2014 already, we did a phase 2 randomized placebo trial at Mass General Hospital, UMass, and the Mayo Clinic. And that will be published very, very soon, within a week or two. But we didn't wait for the publication, of course. We immediately went forward because we saw very strong signals of efficacy in ALS, never seen before. And we have a phase 3 trial, which is powered to efficacy, 200 patients. In addition to the 3 centers I mentioned, we added 3 additional centers in California, Cedars-Sinai, CPMC, and University of California of Irvine. The reason why we did 3 centers in California, the California Institution for Regenerative Medicine gave us a non-dilutive grant of $16 million. And of course, they wanted us to treat more Californian patients because it's California taxpayers' money. So, and we are very happy to announce that since last year I was there at the conference, we were just in the beginning phases of the trial. We have enrolled all 200 patients, and the clock is ticking, just like hair. And in a year from now, in October, we should finalize the trial, and I believe that in November or December, we will publish our top-line results for the phase 3 ALS trial. Just short design, I will go through this fast. I presented it last year, and many of you have seen this data, but this is the data we can discuss, because as you know, the phase 3 trial is a blinded trial, so we cannot discuss any data from that trial. We don't know who was on treatment and who was on placebo. This is the design of the trial is very similar, but the phase 2 trial was 48 patients, was powered for safety, not powered for efficacy, and therefore we did 3-to-1 randomization, meaning treating as more patients as possible, both to be able to verify safety, so the more patients you get more data, and also doctors, of course, when you don't know how to power to efficacy yet, so they want to treat as more patients as possible. So we had 3 centers, and what we amended the trial, we had an amendment after the first 9 patients. So we have an intrathecal injection, so we're able to subtract the CSF when we do the lumbar puncture. But we added an additional intrathecal injection, a lumbar puncture, 2 weeks after treatment. So we were able to compare CSF from before treatment to after treatment, and we were hoping to see a biomarker support to what we're doing, maybe method of action, etc. And we did, and that's one of the very outstanding outcomes of the phase 2 trial that we have seen that is not known in any other ALS trial, where it's very consistent, treated versus placebo in the biomarkers. And I'll talk to that in a minute. So of course, safety profile was very good. Outstandingly, we had no deaths and in the entire trial, no dropouts related to SOEs. And we did have some kind of adverse events, but they were transient, headache, fever, etc., with 24 to 48 hours. So this is a slope improvement, the mean pre to post LS mean slope change per month. So in the left-hand side, you will see all participants, and you see the transient effect. And we pre-specified to look at the rapid progressors. The reason how we knew to pre-specify rapid progressors and why we thought rapid progressors will have a better result, because we saw that in the open labels in the phase 1-2, that the rapid progressors had a better result. And it's a debate between the scientists if it's a biological effect or it's only because of measurement and statistics that it's easier to show a difference. So as you can see, at 2 and 4 weeks, we saw statistical results